In this first part of our series on adaptive immunity, we're going to discuss lymphocyte development. Lymphocytes develop from hematopoietic stem cell precursors in the bone marrow and the thymus, and for this reason, these two organs are referred to as the primary lymphoid organs. B cells mature in the bone marrow. T cell precursors migrate to the thymus, and there they finish their maturation process. Conversely, lymphocyte activation occurs in secondary lymphoid organs, lymph nodes and spleens. This is when mature T cells that have already undergone development in the thymus are activated by dendritic cells to differentiate into helper T cells or cytotoxic T lymphocytes, and mature B cells that have already undergone development in the bone marrow are activated and differentiate into long-lived plasma cells. Lymphocyte development is driven by cytokine survival signals. Hematopoietic stem cells commit to the lymphoid lineage because of exposure to a number of cytokines, including interleukin 7. We know that interleukin 7 is a critical cytokine for this process because in the absence of interleukin 7 signaling, T cell and B cell development is defective, leading to the development of severe combined immunodeficiency, or SCID. This lymphoid lineage commitment occurs in the bone marrow. Also, in the bone marrow for B cells or in the thymus for T cells, committed lymphoid precursors undergo antigen receptor rearrangement, a process termed positive selection, and are eliminated if they are autoreactive to self-antigens, a process termed negative selection. After this, these mature but naive lymphocytes can egress to secondary lymphoid organs, where if they are exposed to a foreign antigen can differentiate into effector or activated lymphocytes that mediate their antipathogen responses. So the first issue is, how are diverse antigen receptors generated? This is a process of DNA recombination. The antigen receptor locus does not have a coding gene. Instead, it is composed of gene segments of various types. There are a number of variable gene segments, diversity gene segments, and joining gene segments, as well as a number of potential constant regions. In the bone marrow or thymus, antigen receptors undergo a process of DNA rearrangement, whereby one of the variable, one of the diversity, and one of the joining gene segments are randomly selected and recombined together to create a coding region that can combine with a constant region to generate an antigen receptor. So this means that there are many, in fact trillions, of possible combinations of gene segments that can lead to an antigen receptor coding gene. That means every B or T cell expresses its own unique antigen receptor. B cells can swap their constant region. Initially, their constant region is the mu constant region, and that's why when B cells get activated, they make the antibody isotype IgM. But after activation, they can receive additional activation signals that can lead them to utilize a different constant region. The process of positive selection is one in which only developing lymphocytes that have successfully undergone this DNA recombination event can actually survive. And negative selection occurs because autoreactive lymphocytes during development, when they see an antigen to which they are specific, undergo apoptosis. The B cell receptor is composed of two heavy chains and two light chains. These are covalently bound. There is a constant region, sometimes referred to as the FC or constant fragment region, as well as a variable region that binds to antigen. This B cell receptor is bivalent, meaning it has two antigen binding sites, and in secreted form is called an antibody. Sometimes this antibody is referred to as immunoglobulin. The T cell receptor also has a constant and variable region, but only has one antigen binding site, or in other words, is monovalent. It is only membrane bound and is never secreted. A few notes about T cell development. T cell development occurs in the thymus. The thymus is composed of a cortex and a medulla. It is a lobed structure. The vast majority of cells within the thymus are called thymocytes. These are committed T cell progenitors that have migrated from the bone marrow to the thymus. And this is where T cells undergo this process of positive and negative selection. This is assisted by thymic epithelial cells. One interesting thing to note about the thymus is that its size is maximal at birth and it atrophies with age. This is a process called thymic involution. And by the time you're in your 30s or 40s, your thymus is essentially gone. Within the thymus, developing lymphocytes express both CD4 and CD8. These are often referred to as double positive lymphocytes. They interact with thymic epithelial cells, and if they are reactive to self-MHC presenting self-peptides of low affinity, then they can be selected to become either CD4 positive or CD8 positive thymocytes. Thymocytes that are developing that are unable to recognize MHC presenting these peptides undergo apoptosis. And in fact, 95 to 99% of thymocytes die at this stage due to a failure to recognize self-MHC molecules. These developing thymocytes, once they successfully undergo positive selection, will migrate into the medulla of the thymus, and there they are exposed to many high-affinity self-peptides. If this T cell, developing T cell, binds to these high-affinity self-peptides, instead of becoming activated, it will become apoptotic and die. If it fails to recognize any of these self-peptides, it then can egress from the thymus as a mature T cell and migrate to secondary lymphoid organs.